All right, so we're going to be starting with chapter 13, which is dealing with chromosomes, the order of genes on chromosomes, and how meiosis plays a role in the inheritance patterns that we saw in last chapter. So Carl Correns in the 1900s, early 1900, was a German scientist, and he was the first person to make a connection between Mendel's rediscovered work and chromosomes. Chromosomes were kind of a new thing being discovered, and Mendel's work came back out, and so Correns was the first person to kind of make that connection. And then very quickly, Walter Sutton followed up in 1902 with what he called the chromosomal theory of inheritance, which was a theory explaining Mendel's results with the behavior of chromosomes during meiosis, how they paired up together, how they separated, how the homologs separate during meiosis 1, etc. Thomas Hunt Morgan in 1910 began working with fruit flies, and everybody was kind of studying chromosomes and their inheritance patterns and how they behaved. And so Morgan worked with Drosophila melanogaster, or the fruit fly, and he bred lots and lots and lots of fruit flies. And over time, in one particular breeding, he developed or discovered a mutant fly. It was a male, and it had white eyes instead of the typical wild type eyes, which is red. In fruit flies, we don't really talk about dominant and recessive. We talk about wild type and mutant type. And wild type is your normal or your typical, and then mutant is the different one. So he thought he would stick to what Mendel did with Mendel's crosses, and so he took the mutant male and he crossed it to a normal red-eyed female, and he was just looking at dominance and recessiveness, and would the traits end up being in the same ratios as Mendel. And all of the F1 progeny were red-eyed, and so from that he kind of went down the Mendel pathway and said, okay, well, red eyes, then the wild type must be dominant to the white-eyed type. So then he, again, did what Mendel did, and he took the F1 females, excuse me, and crossed them with the F1 males and looked at his F2 results. His F2 generation did get both red and white-eyed flies back again. So just like Mendel always got, the recessive trait showing back up again, so did Morgan. However, it wasn't quite a 3-to-1 ratio. I think like 18% of them were white-eyed. But one thing that he noticed that was important was that all the white-eyed flies were male. There weren't any white-eyed females. So then he began to wonder, why was that? So was it something that, maybe was lethal for females? Could they not be produced? So he did a test cross with an F1 female, so a red-eyed F1 female and a white-eyed male. And he did get white-eyed females. So he proves that white-eyed females were possible. And so what that then told him was not only is red, the wild type dominant to the white-eyed recessive, that you can have both white-eyed males and females, but he concluded that eye color must be carried on the X chromosome or the sex chromosome, since it was easier for males to show that trait than the females. So there's his parental generation, his white-eyed male and his red-eyed female. And so all the F1, you can see where all the F1 ended up having red eyes, because again, if you're male, you show what's on the X chromosome. In that case, it was red eyes. Then he crossed the F1 female with the F1 male, and he ended up getting white-eyed males back in the population. No white-eyed females at that point, just white-eyed males. So then he did the test cross and took your F1 generation female, back crossed it with a white-eyed male, and then he did get some white-eyed female showing up. And so eye color then is linked to the X chromosome, not on the Y, but on the X. So that was a pretty important contribution, the whole idea of sex linkage, of traits being linked to sex chromosomes. Sex determination in Drosophila is based on the same kind of sex inheritance as in us. 2 X's, you're a female, an X and a Y, you're a male. And we have the same kind of deal. If you have a Y chromosome, that makes you male. And the X is a single dosage of whatever those traits are. Out of our 46 chromosomes, 22 of those pairs are what we call autosomes. The 23rd pair are your sex chromosomes. So everybody has the same 1 through 22, and then that 23rd pair determines if you're male or if you're female. The Y chromosome, as you can see in the picture, is highly condensed, shrunk down, and therefore, if you inherit a recessive allele on the X chromosome and you're male, you will show that trait because there is no balancing counterpart on the Y. So if you inherit one recessive allele, you show the recessive trait. If you inherit one dominant allele, you show the dominant trait. Typically, as females, embryos are, as all embryos actually are developing, the default gender is female. Because on the Y chromosome, there are genes for just basically maleness that kick in over time, but the original X chromosome that is there begins everybody out basically as female. But the SRY, or the sex determining region on the Y gene, that's what SRY stands for, that gives you your maleness, and that's going to produce secondary sex characteristics and male hormones. And so sex determination varies in a lot of organisms. You know, humans and Drosophila have the XY, birds have the ZW, grasshoppers are the XO, and honeybees are diploid or haploid, females being the diploid of the species. So we talk about sex linkage because sex linkage is an important topic due to the fact that it can influence males more than females, and so you can pass that through a family. So usually we're talking about X-linked recessive alleles because a dominant X-linked allele will behave the same as any dominant allele because it only takes one to show up. But if it's a recessive allele, then and it's X-linked, you only need one to show the trait. So examples of that are red-green color blindness and hemophilia. Just a little bit about hemophilia, right? Hemophilia is a disease where you cannot clot your blood. It affects a protein in a long line of proteins that are involved in the formation of blood clots and platelets. Some forms of them, some form of hemophilia is caused by that X-linked recessive allele. If you're a female and you're heterozygous, you are a carrier, but you're asymptomatic. But basically, you know, if you're asymptomatic, that doesn't mean that recessive allele that you carry isn't doing anything. It is actually producing the, you know, non-compliant defective protein, but because you have a normal allele, that normal allele covers for, if you will, the allele that's producing the defective protein. So when you're a heterozygote, you're a carrier and you're asymptomatic at the organism level. But if you looked at it at the molecular level, you would see both of those types of proteins being made. So this is the royal family pedigree. It's an interesting thing to take a look at when you have some time. You can see all the boys that are affected by that. Luckily, William and Henry down here in generation, you know, 7, they're good. So none of their kids should be affected by it. But now there's another idea of something called dosage compensation. Nature likes to be balanced, and so females having 2 X chromosomes get 2 doses, theoretically, of those alleles. Males having 1 X chromosome only get 1 dose. So that doesn't keep things balanced. So what happens in female cells is that at some point in time in development, one of the 2 X chromosomes will be highly condensed. They will coil up and condense down into and become an inactivated structure. It's very random. So let's say it happens at the 16 cell stage. Those 16 cells randomly shut down either the maternal X chromosome or the paternal X chromosome, but from that point forward, that chromosome stays shut down. And so that condensed chromosome is what we call a bar body. if you are a heterozygous female for some trait on the sex chromosomes, you are referred to as a mosaic, a genetic mosaic, because some of your cells will be displaying the dominant allele and some of your cells will be displaying the recessive allele. So that's why you are a mosaic. That's easy to see in the calico cat. Calico cats can only be females because their fur color allele is found on their sex chromosome. So like in this picture, you can see where the orange fur is activated. That means the black allele was inactivated, and then vice versa. When the orange allele is inactivated in those cells, you get black fur only. So that's how you get a calico cat. So calico cats are really only females. Some exceptions to the chromosome theory. We're talking about chromosomes in the nucleus, right? However, we know now that there is DNA in the mitochondria and in the chloroplast. They contain genes. And the traits that are controlled by those genes don't necessarily follow that chromosome theory of inheritance because they don't go through meiosis. They just get passed to the offspring, typically by just one parent, and that typically is the mother, although there has been some new research out on that. But for now, we'll stick with what we know, which is that the mitochondrial DNA is a maternal inheritance. So no matter if you're male or female, if you isolate your mitochondria and study the DNA, it is your mother's maternal line. Plants, chloroplasts are often inherited from the female plant, but that's also very species dependent.